The overall goal of the following experiment is to discover and identify long-range associated DNA partners. This is achieved by fixation, cell lysis, and digestion of DNA by the first restriction enzyme, followed by ligation to create a chimeric DNA composed of segments from the two associated genes. Next, linkers are added to the chimeric DNA after the second restriction digestion to facilitate its amplification. PCR amplification is then performed using linker and bait DNA-specific primers in order to obtain sufficient amounts of the associated DNA fragments. Results are obtained that show the long-range associated DNA partners with the bait DNA based on sequence analysis of the amplified DNA fragments.
The main advantage of this technique over existing methods like chromosome conformation capture or 3C is that ACT assay can be used to identify unknown long-range associated DNA partners effectively, whereas 3C allows for verification of only previously suspected interactions.
To begin this protocol, culture human cells in an incubator supplied with 5% carbon dioxide at 37 degrees Celsius until they are 80 to 90% confluent. Human promyelocytic leukemia cells are used for this demonstration. Collect the cells by pouring them into a 50 milliliter Nunk tube and centrifuge at 1200 RPM for 15 minutes. Following centrifugation, remove the medium by aspiration. Add 5 milliliters of culture medium to resuspend the cell pellets. After counting the cells with a hemocytometer, take approximately 1 × 10 to the 7th cells to a volume of 40 milliliters with RPMI 1640 medium containing 10% FBS. Then add 1.7 milliliters of 37% formaldehyde to fix the dilute chromatin. Incubate the sample at room temperature for 10 minutes with gentle shaking. Following incubation, quench the reaction with 2.4 milliliters of 2 molar glycine. Next, centrifuge the sample for 15 minutes at 1200 RPM and 4 degrees Celsius. After removing the supernatant, wash the pellet once with 40 milliliters of ice-cold PBS. Then centrifuge the sample to retrieve the washed cell pellet. Resuspend the resulting pellet in 40 milliliters of ice-cold lysis buffer with freshly added protease inhibitors and 0.1 millimolar PMSF. Incubate the sample in a cold room with rotation for 90 minutes. Finally, centrifuge the sample at 2500 RPM for 15 minutes and remove the supernatant and obtain the isolated nuclei. Resuspend the nuclei in 0.5 milliliters of 1x NEB buffer 3 and add 15 microliters of 10% SDS. Incubate the sample at 37 degrees Celsius for 1 hour with shaking. Then add 45 microliters of 20% Triton X100 to sequester the SDS and incubate again at 37 degrees Celsius for 1 hour with shaking. An aliquot of 1 × 10 to the 6th nuclei, or about 15 micrograms, is used for restriction enzyme digestion. To 55 microliters of nuclei solution, add 433 microliters of 1x NEB buffer 3 and 12 microliters of BGL2 to make a total digestion reaction volume of 500 microliters. Incubate the reaction at 37 degrees Celsius overnight. The next day, inactivate the restriction enzyme by adding 95 microliters of 10% SDS and denature the enzyme by heating at 65 degrees Celsius for 20 minutes in a water bath. Add 7 milliliters of 1x ligation buffer and 360 microliters of 20% Triton X100. Incubate the sample at 37 degrees Celsius for 1 hour. Lower the temperature to 16 degrees Celsius and add 50 microliters of 400 units per microliter T4 DNA ligase. Allow the ligation reaction to incubate at 16 degrees Celsius for 4 hours, and then at room temperature for 30 minutes. After the ligation is complete, add 300 micrograms of proteinase K and incubate at 65 degrees Celsius overnight. Then add 5 micrograms of RNase A and incubate at 37 degrees Celsius for 30 minutes. Purify DNA by phenol chloroform extraction and precipitate DNA in isopropanol. Dissolve the precipitated DNA in 150 microliters of sterile distilled water. To add linkers to the chimeric DNA, first digest the DNA by incubating 2 micrograms of purified DNA with 5 units of MSP1 at 37 degrees Celsius for 4 to 6 hours. Following digestion, inactivate MSP1 at 65 degrees Celsius for 10 minutes. Then, precipitate the DNA in ethanol with 1 microliter of 5 mg per milliliter glycogen. Dissolve the resulting DNA pellet in 50 microliters of sterile distilled water. Next, mix 50 microliters of MSP1 treated DNA with 2 microliters of a 20 micromolar linker oligonucleotide L and 1 microliter of a 20 micromolar oligonucleotide S. Then add 1 microliter of sterile distilled water and 6 microliters of 10x T4 DNA ligase buffer. Cover the mixture with liquid wax. Denature the oligonucleotides at 50 degrees Celsius for 1 minute and allow to cool down gradually to 10 degrees Celsius in a 0.5 degrees Celsius per minute gradient using a thermal cycler. Add 1 microliter of 400 units per microliter T4 DNA ligase and incubate the reaction at 15 degrees Celsius overnight. The next day, purify the linker ligated DNA using a QIAquick PCR purification kit and dilute in 50 microliters of sterile distilled water. Choose a primer for the specific region of DNA to be investigated for long-range interactions. In this example, ABLE will be used as the primer or the bait. Prepare the purified DNA for the first round PCR by combining 1 microliter of the purified DNA with 1 microliter of 20 micromolar ABLE1 specific primer 4656 and 1 microliter of 20 micromolar linker specific primer 2963. Then add 3 microliters of 3x ClenTac DNA polymerase 1 cocktail that has been previously mixed with 32p DCTP according to the written protocol, followed by 3 microliters of sterile distilled water. Perform PCR amplification using the hot start PCR thermal cycle described in the written protocol. After the PCR cycle is complete, purify the first round PCR products using a QIAquick PCR purification kit. Elute the DNA in 30 microliters of sterile distilled water. Use 1 microliter of the first PCR product diluted 100 times to perform a second round of PCR using nested primers by adding 1 microliter of 20 micromolar ABLE1 specific primer 4626 and 1 microliter of 20 micromolar linker specific primer 2961. Again, add 3 microliters of 3x ClenTac DNA polymerase 1 cocktail and 3 microliters of sterile distilled water. Execute the second round of PCR using a thermal cycling schedule of 25 cycles of 95 degrees Celsius for 20 seconds, 67 degrees Celsius for 40 seconds, and 72 degrees Celsius for 1 minute, followed by an extension at 72 degrees Celsius for 5 minutes. Visualize the PCR products by running a 5% urea page gel and then scanning the exposed screen in a phosphoimager. Each PCR band can be recycled from the gel by dissolving the gel strips in an Eppendorf tube containing 60 microliters of sterile distilled water and incubating at 95 degrees Celsius for 5 minutes. Centrifuge the dissolved gel products briefly at 10,000 RPM for 10 seconds to collect all the samples. Remove 1 microliter to use as the DNA template for performing PCR with the primer pair 2961-4626 using the same conditions as described above. After purification and sequencing, analysis can be performed with an online tool to determine chromosomal location. Once at the Genome Bioinformatics site, click BLAT to generate a new window and paste the DNA sequence. After submitting the DNA sequence, the BLAT search results will appear. Click Browser on the hit with 100% identity to reveal the location of the DNA sequence. The ABL1 region was used as bait to determine its long-range DNA interactions. 2 BGL2 sites and 1 BAMH1 site were chosen for the ACT assay. In the second round of PCR, primer set 4626-2961 was used to amplify ABL-M1. 4630-2961 was used for ABL-M2, and 4636-2961 was used for ABL-M3. A typical gel pattern reveals one to several bands, as is shown here for ABL-M1, ABL-M2, and ABL-M3. Here, the DNA sequence of clone ABL-M1 is shown to demonstrate that each fragment from an ACT assay consists of 2 combined DNA segments. One segment is derived from the bait DNA region, while the second segment comes from the associated partner. The segments are joined to each other by the first restriction enzyme recognition sequence. The second enzyme recognition sequence will appear at the end of the associated partner sequence. The cloned ABL-M1 fragment contains DNA from the region of ABL1 located at chromosome 9q32.4, and the associated partner is located at chromosome 3p13, which was identified as PROC2. Similarly, the ABL-M2 associated partner was localized to chromosome 5q21.1, while clone ABL-M3 was identified as an intrachromosomal association near the ABL1 locus. In contrast to 3C assays, the methodology outlined in this video will select for the most prevalent long-range interactions. However, by increasing the number of PCR cycles, it is possible to identify additional, less frequent associations as well. Using the imprinting control region, or ICR, at the IGF2-H19 locus as bait DNA in mouse fibroblast cells, different cycling programs were applied in the first and second rounds of PCR in the ACT assay. The ACT assay can also be used to identify differences in nuclear architecture and long-range interactions between normal cells and cancer cells. Normal colon tissue and colon cancer tissue was homogenized, and the ACT assay was performed following the procedures described herein. Shown here is the DNA structure of the ICR region at IGF2-H19 locus and the IGF2 gene. DPN2 sites for the ACT assay are labeled. Primers used in the second round PCR are labeled by arrows and numbers in different colors. The gel pattern of the ACT assay represents the PCR results using primer 2961 with either primer pair 4161, 4163, 5145, and 5151. Unique bands that appear only in normal colon tissue are labeled by yellow arrows, and those bands that appeared only in colon cancer tissue are labeled in red arrows.
After its development, this technique paved the way for researchers in the field of molecular biology to explore nuclear architecture and gene regulation in a 3-dimensional manner.